We're joined today by members of Autism Speaks and by members of the McGovern Institute Leadership Board, by friends of the McGovern Institutes, faculty members of the McGovern Institutes. We have a full range of our both researchers and supporters here. Just a bit about the McGovern Institute. It was founded 10 years ago here at MIT by Pat and Lori McGovern. I'm very pleased that Pat McGovern is here with us today. And it was founded with a dual mission, which is to understand the brain and then to apply that knowledge gained through basic research in the brain, apply it to helping people affected by many different forms of brain disorders. And autism has become a major area of interest for us. Several of our faculty members have taken up research programs in autism. And I'm very pleased to say that the people who have taken up this interest in autism research are all people that began in basic neuroscience, just understanding the absolute fundamentals of how the brain worked. And in the course of their work, they both saw exciting possibilities for applying their knowledge to helping people with autism. And they had the opportunity here to pursue those ideas. We've been aided by many supporters, of course, support from the federal government, but also from many private supporters and foundations, including the Ellison Foundation. And I know that some of the people here today are families that have participated in some of the Ellison supported research studies in the Imaging Center. And you're going to be hearing from Nancy Camisher a little later, who directs some of the Ellison studies, as well as John Gabrieli, who I will tell you about in just a moment. Today, I also want to say just a bit about myself. I'm Bob Desselman. I'm the director of the McGovern Institute, and I spent my almost 25 years of my scientific career at the National Institutes of Health in Bethesda, and when I left, I was the scientific director for the National Institute of Mental Health, and I decided that it was time to leave the government-supported research labs and join the McGovern Institute because I had become completely frustrated by the pace of research that was making progress in psychiatric disease, and I thought that we needed to go both back to basics, go back to the laboratory where people were doing the kinds of studies where new ideas would emerge, and I wanted to take what I had learned to apply to that and to help people see the possible implications of what they were doing in the lab for the clinical research effort, and to see if we can't push that field forward. And that was the best career decision of my life to make that transition, and some of the really exciting work that's taken place in just a few years is some of the work that you're going to hear about in just a few minutes. But now I'd like to introduce Michael Rosanoff, who's here from Autism Speaks. He's the Associate Director for Public Health Research and Scientific Review at Autism Speaks. He's a member of the Autism Speaks etiology team, manages the organization's epidemiology and public health research grants. He's taking the lead in overseeing the International Autism Epidemiology Network and part of the team for the Global Autism Public Health Initiative. Prior to going to Autism Speaks, he was conducting research at Columbia University in neurodevelopmental disorders, neuroscience research, and he brings that background in neuroscience research to this great effort of Autism Speaks to support research directly in this field. So, Michael, turn this over to you.
Thank you so much, and good afternoon, everyone. I just want to start by saying it's truly an honor for me to be here speaking at MIT and the McGovern Institute. In fact, as many of you probably saw, I crunched my way in here and I made it slowly up to the mic. Not even the most severely of broken pinky toes could keep me from talking at MIT today. So again, it is really great to be here. So today I'm going to talk about a subject that's been in the media a lot lately. You can't turn on the news and hear a report about autism and not hear them talk about autism prevalence and how it's increased over the years. When my cousin was diagnosed with autism 22 years ago now, nobody knew what it was. Even when we heard that he was diagnosed with autism, we didn't know what it was. So even within my family, it was just a completely new term, a new disorder to us. But today, 20 years later, it would be very difficult for you to go out and ask somebody if they had not heard of what autism is. I think if you were to ask somebody if they knew somebody with autism, chances are they would say yes. And so I'm going to talk about that today. And what we've seen in the recent literature is that autism affects 1 in 110 children in the United States today. These are numbers published by the CDC. That's about 1% of the children in this country. This also represents a 57% increase in just 4 years of data collection. You may remember it was 1 in 150 before it became 1 in 110, and that is quite an increase in a short amount of time. But if you look at the data over 20 years of research publications, you actually see that autism prevalence has increased about 600% over 20 years. Again, these are the numbers published in the data. So about 20 years ago, it affected approximately 1 in 2,500 children. And again, today it's almost 1% of the population. But the question that we keep hearing is this a true rise in autism prevalence? Is it a true increase? Is it real? So what does this mean? And that's what I'm going to talk about today. Some of the new research coming out of the United States data, and California in particular, that tries to decipher what this increase in autism prevalence really means. And so one of the studies that came out showed how actually over time the change in diagnostic criteria for autism may have accounted for some of this increase. More specifically, it was the change in diagnosis from mental retardation to autism. So those two circles, this is taken a little bit out of context, but this picture appears in the report. And what it shows is that that overlap is the new autism cases actually resulting from that shift in diagnosis from mental retardation. And to look at it a little bit differently, what you can see here is that dark slope at the bottom is the expected change due to the expected change in autism prevalence over time due to that shift from mental retardation diagnosis to autism diagnosis. But what we saw here was that extremely large increase, so that gray area is the unexpected rise. And what the researchers from this investigation said was that about 26% of the increase in autism prevalence over that time period in California could be attributed to broadening diagnostic criteria. Also coming from that research lab, what we saw was this idea, we've heard of this idea of maybe the rise in autism prevalence could be due to an increase in awareness of autism or better understanding of autism. And so what this paper demonstrated was that actually the proximity of a child living to a previously diagnosed child with autism actually increased that child's odds of being diagnosed themselves. So the theory here is that there's some kind of social mechanism taking place where there's this information diffusion or sharing of information within the community that's actually increasing a child's odds of being diagnosed with autism. So in a way, it's saying that awareness is contributing to the rise, and the investigators in this study estimated that 16% of that rise in autism prevalence could be attributed to this increased awareness or information diffusion within the community. Recently in the literature, we've heard about parental age being a potential risk factor for autism. Parental age, we do know, is a risk factor for birth defects and other neurodevelopmental disabilities. But in terms of autism, we're seeing consistently, again, that parents over the age of 40 are at significantly higher risk for having a child with autism than their younger counterparts. And you can see here the two the gray and the black lines actually are the father's and the mother's age, the risk for fathers and mothers over the age of 40, whereas that white bar is the average risk among the population of parents actually 25 years of age or younger. And so what these investigators came to the conclusion was that 11% of that increase over time, of that slope that we saw, could actually be attributed to this increase in parental age as a risk factor. Now, the mechanism for that is still something under investigation. It may have to do to genetics, but the idea is that in general, parental age is a risk factor and it can account for 11% of the increase. And parental age is a social component, right? Parents are choosing to have children later in life, so it's a social factor that's contributing to the increase. So just to review what I went over and what the research literature is showing us today is that, again, 26% of the increase in prevalence can be attributed to changes in diagnostic practices, that broadening criteria. 16% is due to greater awareness, and 11% is due to increase in parental age. Well, if you add that up, there's still about 50% of that increase in autism prevalence that is due to unknown factors. And so the question is, of course, well, what then is causing the increase in autism prevalence? Well, I wish it was a simple answer, but it's not. What we do know is that genetic susceptibility is likely involved in most cases, but then environmental factors also contribute to one's genetic susceptibility. And so what we know is that it's a complex interaction between genes and environment that are likely causing the rise in autism prevalence, but we need to identify those factors and understand exactly how they're contributing to the increase. So I'm going to talk a little bit about what Autism Speaks is doing to help understand this. And one of the things we did recently was with the Centers for Disease Control and Prevention, we held a workshop on February 1st in Atlanta that actually wanted to look at this change in autism prevalence over time. We wanted to identify promising directions and priorities in the research, and there was just a general need among the community for understanding what this increase means. It's very likely that new prevalence estimates will come out this year from the CDC. I don't know what the numbers will be, but there's a high likelihood that those numbers may be higher. So it's about time that the federal government was able to take a position here and hold this workshop, and we were happy to help them bring together the right scientists, advocates, and individuals with autism to serve as expert panelists in this discussion. And the key outcomes were that we need new analytic models for understanding how these multiple risk factors actually work together to increase one's risk for autism, how gene and environment work together. Also, there's a need for better defined autism phenotype data, and that is the autism spectrum disorder is a broad spectrum. It's often said that no two cases of autism are exactly alike. So there's a need for better phenotyping, better characterization of the cases in the data. And then, of course, data quality, it just in general is, of course, key. Autism Speaks is also funding research on all of these areas that are appearing here. And to date, Autism Speaks has committed over $160 million to autism research through the year 2014. And just to give you an idea of where we're funding some of this research, I picked the greater Boston area for obvious reasons, but 32 grants from the greater Boston area from 10 institutions are currently being funded at about $7.5 million from Autism Speaks. And you can actually learn about these grants. This is a little plug for our brand new grant search. It's grants.autismspeaks.org. But what I did here was I wanted to check out what we were funding at MIT, and there are two projects that we're funding here that you can see below. Autism Speaks is also interested in understanding what prevalence is like around the world. Right now, the research suggests that about 1% of the global population has autism. However, there have been no studies conducted outside of these high-income developed countries that you can see in blue. We have no idea what the autism prevalence is in the developing world or even middle-income countries. So what we're doing is we're working with investigators and exploring opportunities to look at what is causing the rise in autism prevalence in over 30 countries around the world through a number of our programs, our International Epidemiology Network and our Global Autism Public Health Initiative. And the reason why we're doing this is because international research creates unique opportunities to investigate genetic and environmental factors. For instance, maybe in some countries there are environmental factors that are common where they're not so common here in the United States. At the same time, there may be certain populations that are very genetically related, and there may be opportunities to find rare genetic mutations among those populations. In addition, it gives us a place to study creative diagnostic and intervention strategies among underserved populations. Even here in the United States, children are not getting the services that they require and that they need. So by being for example, if you were able to train parents, for instance, in how to provide intervention in the home, we may be able to use that as a model not only in the developing world where there aren't trained professionals to do that, but also here in the United States where people don't have access to the appropriate services. So what does it all mean? Well, the autism crisis is real and growing. It's clear that regardless of whether or not autism prevalence has increased over time, 1% of the population is huge. There are more children today diagnosed with autism than ever before. And half of that increase that we saw over time is still unexplained. It's not caused by just better diagnosis, greater awareness, and social factors alone. There are genetic and environmental factors at play, and we need to understand those environmental factors. And to do that, Autism Speaks has committed $160 million in research funding, but we still need answers and a better response to this public health crisis. We still need more funding to do this work. And just to give you an idea of what I mean by public health challenge, autism prevalence, as I said, is 1 in 110 in the US. Type 1 diabetes, 1 in 400. Childhood cancer, 1 in 2,000. Cystic fibrosis, 1 in 3,500. So this year, more children will be diagnosed with autism than those other conditions combined. And the annual costs in the US are an estimated $35 billion to society, and for each individual with autism, about $3 million over their lifespan. So we need better detection, effective and affordable care. There's a growing adult population in need of support. The costs to society, as you see, are immense, but the costs to families themselves are immeasurable. And so just to conclude, a call to action, 1 in 110 children have autism in the United States today, about 1.5 million individuals, many of which are underserved. What we do know is that early diagnosis and early intervention are the keys to improving long-term outcomes and lifetime costs in autism. And additional research funding is absolutely essential to finding the answers to the increase in autism prevalence and to provide services to the millions affected by autism in our country. And so with that, I just want to say thank you for your support. And again, it's a real honor to be here, and I look forward to the question and answer period. Thank you.
Michael. So we now have 2 short faculty presentations, and that will be followed by an open discussion. And we're keeping the faculty presentations short so we can spend more time in the discussions, let you have a chance to ask questions and really engage people. And Nancy Canwisher will be leading that discussion. But our next speaker is Guoping Feng. Guoping is someone who has established many of the most important phenomena of what we know about the synapses. Synapses are the connections between neurons. It's how neurons communicate. And he had spent the early part of his career working out all the different proteins that interact in the synapse. And in the course of doing that work, he made some really exciting discoveries that led to an animal model of autism that was just published in Nature. Rather than go on and on about this, I will let Guoping tell you about this himself. But I did mean to say that Guoping is the Poitras Chair of Neuroscience in the McGovern Institute. And it's a chair that was established by Jim and Pat Poitras, who have been very strong supporters of research in psychiatric illness. And Jim and Pat are also here today. So, Guoping.
Welcome to McGovern. So in my lab, we are interested in how our brain build the machinery for communicating between neurons. So this is basically the building block of our brain function. We have billions of neurons in the brain, and then we have trillions of connections. Our brain communicates in a very precise way. So that's why we can think and doing things very precisely. And so you can imagine that there are many different ways to build a connection, just like a wire computer. In neurons, they communicate through this very little apparatus called synapses. They're basically 2 neurons linked together. They have a structure, and they have hundreds and hundreds of proteins actually to help them to do their work. So each little synapse is about a couple microns, so very, very small. But there are over 1,000 proteins clustered over there to do the function. So if there are so many proteins, it means that these are very, functional, very diverse. They do a lot of things, but it also is a trouble spot, right? So any of these proteins goes wrong, genetic mutations or whatever the factors, we just heard, could cause a problem for neuron-neuron communication. So recently there are a lot of human genetic studies from patients suggest that the evidence converge onto one of the biggest factors that could contribute to autism is synaptic dysfunction, because many of the genes mutation found, they all many of them play a role at the synapse. So in my lab, we study how synapses develop and how they function, how they are modulated. So during that course, and we found that their genes are critical for the process of synaptic development, and if we disrupt them, if there are mutations, that will lead to a dysfunction of synapse, a dysfunction of neuronal communication. Now, these in humans can cause psychiatric disorders. In mice, we cannot diagnose them autism or OCD, but we can diagnose them with phenotypes, their behavior similar to some of the phenotypes of autism or OCD or bipolar disorders. So we are particularly interested in this is a synapse. Can you see one neuron communicates with another neuron? So we are particularly interested in a group of proteins that in this side of the communication, the receiving side, we call the postsynaptic side of the synapse. And so some of the proteins have been implicated in several psychiatric disorders. And I'm going to focus on one protein, which there are very strong evidence, human genetic evidence, called Shanks. Oops. Let's see whether. So this protein is basically. My mouse disappeared. Here we go. So this is the protein which is linked the important proteins, ion channels from membrane from the synapse to the membrane, so to the cytoskeleton. So particularly we are interested in this is called on the right side called Shanks3, because this protein, for reason we study them, because there are many line evidence suggests that in one particular brain area called the striatum, which is in the middle of the part of the brain, and well, I will use you can use this. Can we use this? No. Okay. So in this dark part of the brain, which is the striatum, so it's highly expressed there, and the dysfunction has been implicated in many psychiatric, especially in OCD and autism. Oh, thank you. And so this part of the brain. So they are also, while we're studying this, try to understand how this protein functions as a synapse in the neuron-neuron communication. There are quite a few clinical studies show this gene, actually mutation of this gene is very strongly linked to autism. There are actually hundreds of patients with either deletion of gene or mutation of this gene. And so there is a large-scale study very recently published that suggests there are about 0.7 to 1% of ASD patients actually contain this mutation in this gene. So it's a very significant factor because current study have not identified a very significant common allele for any of the psychiatric disorders. So we thought this is the most likely a monogenic factor, and many others may not be monogenic, but by studying a monogenic case, it will help us understand what is really wrong in the brain of these patients, then can help us to understand what is the mechanism and how can we correct this. And this could have, if we understand this mechanism, it could have a broad impact in understanding what other autistic patients may have in their pathological brain. So our study is by generating mutant mice. Right? Autism, you all know, have three major core symptoms: repetitive, compulsive behavior, sometimes self-injurious, and they have language impairment, and they have social interaction deficit. Of course, it's very hard to study language. I don't know whether they speak English, Chinese, or French. I don't understand, but there are things we can study in mice, right? For example, repetitive behavior. So when we mutate this gene, they have repetitive compulsive grooming. They're grooming their skin and hair and skin, they're still grooming. So self-injurious grooming. And most importantly, they have mice are social animals. You can study their social behavior actually very easily. So these animals actually, they don't like social at all. These are scientifically, I can show you a video that they don't like actually social with other mice. So I'm going to show you two videos. This is a video and showing, just put them, this is one. Just put a few mice in a cage. Let how they interact with each other. So this side is the Y type, just regular. This is a mutated with SHANK3, and you will see when. Now I need to find the mouse, otherwise. So you can see that when you put mice together, right? So initially they will, pretty soon they will get together to interact. So this is the mutant mice. These are white-type mice, and so in the white-type mice, they start to sniffing and start to play. Right? You look at these mutant mice; they always stay by themselves. Do whatever they like to do. This mouse will stay there digging for all the time. Right? Eventually, these white-type mice, they will start interacting with each other, with sniffing and play around. I don't know what they're talking about, but they are playing. Right? So these, you can see the whole time you videotaping them, they actually never get together. They will do somehow they are active doing something, but they are repetitive doing something and just not interested in interacting with other mice. So, and the next video I show you is that if you put a novel object into this is also happening in humans, and they are afraid of the novel object. So this is, you basically put a new toy in their home cage, right? So this time, this side is the mutant mice, this side white-type mice. So when you put this in, and you will see very peculiar phenomena. So this is white-type. You can see they go passing by, and eventually they will sniff and go on. This, you see, they are very scared. You're getting close, they will startle back and just never touch it. So this, eventually, you will see they sniffing and they'll start getting play the new toy. So these mutant mice, you can see they running around them and look at them and really scared of. There's nothing, it's not hard or cold or anything. Right? It's just a new toy. So tell you these mice actually have a phenotype of afraid of phobia of novelty. So the same thing for novel mouse. You put a new novel mouse in, it will completely avoid it. So these are phenotypes that we think we can characterize. So the key thing to a neurobiologist try to understand this type of behavior is why. Why you mutated a single gene would have in these mice have this peculiar deficit, which is resembling some of the phenotypes in symptoms in autistic patients. So the idea is in animals you can do a lot of advanced studies, use biochemical study, electrophysiological study. You can look at brain activity, look at communication. So a particular area we're interested in our communication between cortex to the striatum, which is a very area strongly implicated in autism and OCD. So when we look at them, indeed, the neuron-neuron communication is very severely disrupted. So we think that we have other evidence such as this is the key this defect is the key to the repetitive behavior compulsive. Because if you put a gene back only in that area, you can actually rescue the repetitive behavior. So, and the interesting for autism, for this autism model is we also see structural defect in neurons. They actually have a much complex neuronal structure. And their neurons are more complex. They have more processes. So that got us interested because in human patients, you're probably very clear, aware that one of the most robust findings in autistic kids is they have an enlarged brain. It's not in every kid, but it's very significant. So actually, we did a 3D volumetric structural MRI study to show, we show that in these mice, in the mutant mice, they are striatum, they are caudate, is also enlarged. So it's similar to what we found in human patients. So suggesting that some of the neuronal structure change in the brain at the individual neuron level may contribute to this phenomenon we found in these mice. So the idea is that by studying this, we can understand what structure is defective and what type of defective. That'll give you a way to find an approach to correct them in mice first, and hopefully that will lead us to in the future to test these hypotheses in patients. Right? For example, we don't know what causes social deficit, but there are many theories. Not with these mice with such a robust social deficit, we can test them. So in my lab, we're testing how can you characterize the social deficit. Which part of brain region actually responsible? So we can put a gene back or use chemical ways, use drug to correct this problem. Can we reverse this social deficit? So this kind of study will help. Hopefully, will help us in the future. Although we are doing in a lab, in a very, very basic research, and use animal as a model, we're hoping by combining human genetic studies with the gene mutated known cause autism in human patients, will help us to find a way to improve our understanding of hopefully in future develop a new strategy for treatment. So that's, I'm going to stop here. These are the people, I didn't do any of this work. These are the people who did my work in my lab. Thank you.
we're going to hold questions to the general discussion, which will be shortly. So the next person presenting is John Gabrieli. John was recruited to the McGovern Institute from Stanford and is also the head of our imaging center, which you will be invited to take a tour of at the time of the reception. John is another person who started out in his career studying the basic biology of the brain, how neural circuits support memory formation, and as he began extending his work into child development and emotional development and so on, he took up an interest in autism disorders, and he's going to be telling us about some of the work that they're doing in human subjects in the imaging center as part of the autism project. John.
MIT can solve any problem except the switch from Apple to a PC. Yeah. Thank you. I know, now you're going to be disappointed. Sorry. Sorry. Thank you. I have one, another one. Yeah, thank you. Good, thank you very much. Okay. It's a pleasure and an honor to speak in this program. It's a tremendous privilege to do research at MIT and to work in the McGovern Institute for Brain Research. The scientific inspiration of my peers, the technical support through the remarkable, in my area, imaging center downstairs, which together the MEG Center, the MRI machine, ERP resources really make every major method we have for non-invasively looking at the human brain available to all researchers in a small space, comfortable for research participants. It's really a huge privilege, and the philanthropic support that helps us advance at a much greater rate than we would otherwise. It's a huge privilege and a huge responsibility. So I'm going to tell you a little bit about some of our research recently on looking at, in humans, at brain disorder aspects of autism. And we're all struck by I think, those of us who work on typical brain functions as well as difficulties in brain functions, about the fragile power of the human brain, the capacity to feel, dream, desire, remember, and move, socially interact. On the other hand, every one of those powers has an Achilles heel in the brain, a risk. You know, when you see 1,000 proteins sitting around in one tiny synapse, as we heard so eloquently before, it's so amazing on the one hand, and it's so risky on the other. And also the sense that I work on developmental disorders, the neurodevelopmental lottery that every infant and child and family participates in, the large number of individuals who will be born with something that will make life challenging for them, and something that we want to do better about to help those children flourish and families flourish. And so, as you heard and you know, autism is typically defined as involving 3 areas of difficulty or difference in social interaction, in communication, and in repetitive behaviors. The research I'm about to describe today is on individuals who would have the diagnosis of Asperger's or higher functioning autism. That line is about to be erased in DSM. There's going to be all kinds of discussion about what that means or doesn't mean. On the one end, and it's going to be erased on the other end, supposedly for PDD-NOS. And we can discuss maybe what we think about that, but it sort of broadens the spectrum and what we understand to be a spectrum of disorder and difficulty. For the kind of research I'm describing today, all of our research tends to focus on individuals who are stronger cognitively in language, because They need to understand instructions, go into scanners, and we're all acutely aware that in many ways our research efforts underserve individuals who are, for example, non-communicative, and that's something we think about a fair bit as well. And I have tremendous thanks for the research participants and families and for the initial sources of support that let this research get done. And we're also aware that individuals and families dealing with autism, they have a full and challenging life, and to take the time and effort to come and participate in research, we're very grateful. None of this would happen without that kind of participation. So I'm going to focus on a behavioral finding and a couple of findings of using brain imaging, and I'm going to focus on studies about an experiment, a theory of mind. And that's the ability to attribute mental states, such as beliefs, desires, and knowledge to oneself and others, and to understand that others have beliefs, desires, and intentions that are different from one's own. So it's basically the capacity to imagine that other people's minds can have different perspectives, are full of feelings, thoughts, and plans, and that they can differ not only from our own thoughts and feelings, but perhaps even from the physical reality around us. And it's been known for quite some time the development of theory of mind. So a child becomes sort of adult-like around 6 or 7 on many of the kinds of tasks where they have to think, including, by the way, deception. Not all of theory of mind is delightful and about understanding other people, but knowing how to deceive other people depends upon understanding what they are willing to believe is possibly true. So it's the full complexity of human relations are captured in the understanding of what other people think. And that for many years it's been shown that adult-like theory of mind is delayed even in individuals with high-functioning ASD, 5, 6, 7 years, depending on the measure. But behaviorally, there's been kind of a mismatch, which is that in high-functioning adults, adolescents, and so on, the kinds of tasks developed for children are successfully performed. And so you have a mismatch between the kinds of everyday, practical, real reports that families and individuals tell you, which is it's a bit challenging to talk to people. I don't understand really where they're coming from. I might say the wrong thing. I don't know what feeling they have and how to best address that. And doing very well in these tasks of theory of mind. And that's because many of these tasks were originally developed, in fact, all of them to start with, were really originally developed for young children. That's where the literature came from originally. And so a high-functioning adolescent or adult knows the correct answer, and we can't study in science what we can't measure. We can't begin to analyze and understand something if we can't measure it in the laboratory. So we want to be able to measure in some way experimentally this challenge in understanding or this difference in understanding what's in the minds of somebody else. And so together with Rebecca Sachs, Leanne Young, and Joe Moran, we looked into the realm of moral judgment to kind of develop what you might call a stress test, a more challenging test of thinking about what's in the mind of somebody else and how you value that or weigh that in your own analysis of what's going on. So we focused on the difference between intentional harm, when you mean to harm somebody, and accidental harm, when harm occurs but you didn't mean to do it, and failed attempts. And so we kind of put a stress or a competition between two things, and you'll see the concrete example. The outcome, whether something good or bad happened, and the intention, whether you meant something good or bad to happen. And by putting those things in opposition, for example, accidental harm, innocently you do something, but something bad happens to somebody. Well, now we put them, there's no bad intention, but there's a bad outcome. So individuals read a series of stories would have roughly this property. Sometimes they would be told that an individual thinks powder is, for example, is toxic. It is toxic. You give it to your friend and she dies. These are very rough stories, but they're just little stories. And everybody agrees, Grace was bad. Okay, bad is morally impermissible to do that. If Grace passes sugar, the sugar is fine, nothing happens. Everybody says there's no moral problem to be weighed here, no problem. The two situations where we put intentions and outcomes into opposition are these other two conditions, where Grace thinks the powder is sugar, it's toxic, and the friend dies, so she passes the sugar too. So, okay intention, bad outcome. Or, bad intention, she thinks it's toxic, she passes the sugar, but nothing bad happens. So see, in these two cases, there's kind of a stress or tension between the intention of the person and the outcome of the act. And then we asked, what happens as people rate the moral permissibilities of these actions? So in this graph, the higher the value is the more you think it's morally permissible to do things. And the first thing to notice is that these high-functioning individuals with autism spectrum disorder, pretty much look similar in most cases, as you might expect in making these judgments. Nothing, if something very bad happened and it was intentionally done, everybody agrees that's very bad. Nothing bad happened, everybody agreed, no problem. Where something happens is here. So typically developing individuals, neurotypical individuals, forgive accidental harm. And keep in mind that something bad happened, right, in this story. But they say, well, if you didn't mean it, it wasn't so bad. Individuals with a diagnosis of autism don't forgive it so much. There's not a right or wrong answer on this. I mean, these are stories in a laboratory. You're telling a story. If it's my relative and somebody accidentally does something terrible to them, I'm not sure I'd be as forgiving on the intention. There's not a right answer to this. There's a weighing of how to think about a complex moral situation. And so a chance for us to be able to measure the weighing of outcomes versus intentions, and intentions reflect theory of mind, right? It's understanding what a person meant. That's a test or direct measure of that. So we're hoping this will help us have a way to better measure differences in how people approach thinking about situations, and then be able to examine whether, for example, certain kinds of treatments affect this early on that might be helpful to individuals. Let me switch to two results from brain imaging. And in this experiment with Elizabeth Redkay, who's now on the faculty at Maryland, we looked at the difference of what happens when you look at somebody who's looking directly at you versus somebody whose gaze is averted. And as you know, if you talk to somebody, it's a very different social experience if they're looking at you, or if while you're talking to them, their eyes are over here, their gaze is averted, right? You know that they're paying attention to something else. It's a different social communication. And so we had adolescents with or without the diagnosis of ASD see in different conditions a face looking right at you or a face looking away. And again, the face looking at you we think is, that's a person who's ready to do social communication with you. An averted face of somebody who's towards you, suggesting they're shifting their attention somewhere else, something's more interesting, they're not interested in you. It's a different social cue and a very fundamentally social cue. What we found in typically developing adults is two brain areas that responded in opposite ways to where the gaze was directed. So that if they were looking at averted gazes, activation in the posterior part of the superior temporal sulcus. The posterior temporal sulcus is a big brain region, but it's been implicated in social cognition, thinking about how people are talking, looking, moving their mouth, lots of aspects of social cognition. And you might imagine that what's going through your mind if you're talking to somebody and they're looking that way, right? You're thinking, why are they not listening to me? What's going on, right? So this is some sort of a response to that antisocial signal you're getting from somebody. In contrast, in the insides, in the medial prefrontal cortex, a region involved in often responding to how other people are thinking, more response the opposite direction when somebody's looking at you than looking away. So we don't really know what this means until there's, how to think about this in a deep way, but, perhaps, this is a part of the brain that gets engaged when you're thinking you're having a direct social interaction and some exchange of mental pathway or sharing of thoughts. And when we look at individuals in the same age range, very high functioning, very smart individuals with a diagnosis of autism, here's what we see. Minimal difference in the posterior cortex and also minimal difference in the medial prefrontal cortex. How to interpret that, is a long step to go, but much less sensitivity as far as we can measure in the brain for differentially responding to these two kinds of social cues, both the eyes towards you and the eyes averted. I'm going to present two more slides with two more results. One of the striking things in the literature, again, this is always with individuals who are very cognitively functioning well, is this, which is an observation that in the laboratory, again, sort of like with the theory of mind, that people do very well on these kinds of tests we give them, and then they say, yeah, but in real life it's really challenging. And one of the things that happens in the experiments we do in the laboratory is we've unusually sort of well-organized, formatted conditions. You know, we say, get ready, you're going to do X. That's not like life where you're walking around, things happen, right? We don't say, get ready to be social. Okay, you know, so because that's how experiments are. They're not like real life. And so, on the other hand, we have to, you know, that's how we do science. So one thing that's been noted is this, and there's, you know, this is built on work from Nancy Canwisher, who discovered the fusiform face area, the first area in the brain that selectively responds to faces compared to all other visual stimuli. And some researchers have said, well, it's kind of a messy literature on what happens in this part of the brain, and complicated literature. But some people have said, well, if you can, you know, sometimes we get less response in this area, and the data is all over the place, actually, when we show faces. But if we put, and this is not our work, this is work from Boston University, if we put a red dot to draw the attention of the individual to the face, to focus them, you know, by this little manipulation, then we don't see any difference between typically developing young adults and adults with ASD. And they said, well, when we see differences, maybe it's because what spontaneously individuals with ASD tend to do, but if the stimulus draws them in, then the response is similar. So we just wanted to ask, does that seem correct, okay? Because that experiment involved, one experiment had, some experiments had faces without any red dot. One experiment had red dots only, okay? And we went on to say, in the same individual, if you present something that draws your attention to the face versus another condition where you more freely can attend to it or not, does that make a difference? And we looked in this fusiform face area in the posterior cortex, and here's what we found. For typically developing adolescents, it doesn't make a difference. That is, they're interested in faces, and it doesn't matter whether the dot is there or not. Faces are very socially interesting stimuli for most people. For individuals on the spectrum, it makes a measurable difference. And we're only partway through the study, but it looks pretty clear. That when the dot is there to draw the attention, then the response is very much like that of the typically developing control participants. But when it's not there, there's less activation, and in some sense, maybe less attention is drawn to the face. So we're not completed with the study. We know this is the final thing. But it's something interesting, because I think what's fascinating about it is, you know, if a red dot can do that, then there's got to be ways that we can help people. You know, then they have the resource available to them. How should I put it? First, there's some things where the resource is not available to people. But if a red dot can make a difference, you know, the right kind of support might make people find it much easier to have social interactions if that machinery is available to them under the right context. And the last result I want to share with you is where we show people faces, neutral faces or emotional faces, and looked in the structure called the amygdala. It's a structure that's in the limbic system and responds to signals of threat in the environment or sources of risk in the environment. And here's what we found, and there's a small twist in the story. So if we looked in the amygdala at the response for emotionally provocative faces, anger or fear, we saw this kind of activation in typically developing young adults and the absence of that activation in individuals with ASD. And you could stop right there and you could say, well, the amygdala is you know, not responding to faces in some way. It's another aspect of the social processing difference in the brain. By the way, we don't see a difference when we look at responses to houses. So it's a social thing. It's not everything. But we can do one more thing, which is not just look at the difference. So this brain activation is a difference between the emotional faces and the neutral faces. We can sort of look at how the responses are in each condition. And what we see is this, that typically developing individuals have a stronger response for these emotionally expressive faces that signal threat, anger, or fear than neutral faces. That's been found many times before. When we look at the individuals with ASD, what we find is they're sort of activated or turned on for both kinds of faces. So you don't see an activation here because this is a difference between the two. And there's not a difference between the two, but what appears to be happening is the individuals with autism spectrum disorder are responding in the amygdala as if the neutral face were as provocative as an angry or fearful face. So when they get the emotionally expressive face, there's sort of no more to go. They're at the top of their scale already. And that might go with the sense, I think, that individuals with autism sometimes feel by their report, and you know, a sense of stress in dealing with people. And it does, you know, for people without autism, the stress mostly happens when people are looking angry at you or scared at you. That's when you figure something wrong is happening. But for an individual with autism, perhaps, you know, even the neutral faces are already triggering a sense of anxiety. that would be, make it less desirable perhaps to interact with somebody. So I always end with a just so story like this. We don't know that's the story. We just know the brain signal. But we want to think about how these brain signals might relate to the everyday experience of individuals. So thank you very much.
So we're going to now we're going to have our discussion, and then that's going to be followed by a reception outside. We invite you all to join the reception. And I also want to point out that this is part of a series that we've started in brain disorders in the McGovern Institute. We just a few weeks ago we had one of these sessions on depression. Next fall we have one coming up in Alzheimer's disease, and we hope that you will stay on our mailing list, take our literature, and if you have registered and allow us to contact you, we'll send you notices about upcoming events, and we hope that you'll come to other events in the future and become involved with us. I'm asking Nancy Canwisher to be the moderator of the next session. Nancy, as John mentioned, was the person who discovered the fusiform face area in the human cortex. And early on, Nancy noticed that, believe it or not, our brains are not all identical. And that if you even just take a simple thing like where is the area that processes faces in your brain, it actually shifts around from person to person. And she became a proponent of the idea that you really have to study each person individually, their brain, and not just average across large groups of people and assume that everybody in a big group is the same. And she took this perspective into autism and said, we really need to understand the whole autistic population at an individual level, because there's almost certainly going to be different subgroups of people with autism that are different, and that we shouldn't just be all averaging them together. And she's initiated a major project in children to understand that individual variability and to try to uncover the fundamental features that are shared by small groups of kids on the autism spectrum. And so she will both moderate the session, but also she is another expert in this field, and she will answer questions as well. So Guoping and John, do you want to come up here? And Michael, are you going to join us up here? Are you willing to take questions?
Sure, of course.
And I'll ask Nancy to be the master of ceremonies. Should have you hobbling up here.
Great. Well, welcome. It was. I don't know if this is on. It was wonderful to hear from all of you, even though I've heard from two of you many times before. There's always new, exciting stuff, and it was also really lovely to have Michael's introduction to the importance of this problem. So, so just to get things rolling, I'd really like to get questions from the audience, but I'll just start by pointing out what's probably already obvious to most of you, is the incredible fascination and challenge and importance of studying autism. So beautifully underlined by Michael's presentation of the fact that the incidence is skyrocketing, and that only some of that increased incidence is attributable to understandable factors like changes in the diagnosis, raising this real importance and challenge of understanding what's going on to change this incidence. But you can also see the fascination and challenge of the huge breadth of different levels of analysis that we have in autism research, with Guoping's magnificent work on the SHANK3 protein, one of those 1,000 proteins at the synapse, and in this incredibly intricate structure in the brain, bridging all the way from there to, in humans, the very high-level phenomenon of the triad of symptoms in autism, and the kind of phenomena that John talked about with specific social disorders. So to me, one of the real challenges is what kind of causal explanation can we come up with that bridges from a particular gene or a particular protein in the synapse all the way up to a high-level social deficit in autism? How do you go from a gene to a very specific social disorder? And that's just scientifically, in the abstract, it's a magnificent mystery. And in the case of autism, it's a pressing need to understand that causal chain. And of course, we need to understand that causal chain in order to understand how to intervene in that system. So I guess I'd like to invite questions from the audience.
I think maybe we should turn off the projector and turn
Okay,
up
do you know how
the
to do that?
lights just for the no, not completely. Let's see if I can get this. All right.
Okay, thank you.
A little more room.
Don't lean back on the next one. And then how do we, do the, there we go.
Okay, wow, all right. We've met. Was it, yes?
First question.
Excuse me, hold on. Let me just hand you a microphone.
Oh, okay, thank you. A non-scientific question. I concur. There's been an incredible increase in the diagnosis of autism in the last 20 years. And has there been any studies at all done as far as correlation between socioeconomic changes? We have gone in the last 50 years into a nuclear society, among many other things. And how is there any correlation at all from having gone not only from an agrarian to an industrial, but from industrial to a nuclear society?
So, is this on? Can you hear me? Okay. I'll just hold my tie up to my mouth. So there has been some research on the influence of socioeconomic status on autism risk. And it does show that higher socioeconomic families do seem to be at higher risk for having children with autism. But the question that comes out of that is it because those families have greater access to diagnostic and treatment services? And so are they being captured in those research studies more often than those of lower socioeconomic status who may not have access to, again, diagnostic or treatment services? So the research there is not definitive. But the parental age stuff that's coming out is a little bit more. I mean, it's been repeated. And what we're also seeing among those groups is that families of older parents of children with autism are also of higher socioeconomic status on average. So there may be a relationship there in terms of the social factors working together that increase the odds of being diagnosed with autism. It may not be a true risk in the sense, though, of an exposure, for instance, that's causing it.
I was just curious if some of the mutations that you saw in mice can be seen through like MRI or some other testing, like the protein mutation or the caudate mutation.
Yeah, that's a good question. So for the structure MRI, you can do almost identical things as in humans and in mice. For functional MRI, John just showed us, it's possible. We are working on them. The resolution is still much lower because the mouse brain is very small. So getting higher resolution, there are different ways to get a goal. So yes, it is possible to do. Actually, we are planning to do it. We just discussed it because it's a very high magnetic field MRI machine down in the basement. So we are in the next few months we're actually going to do that. But the resolution is still much lower. So you cannot say, okay, we're not totally sure you can, for example, amygdala in John's picture is pretty big. That's almost as big as a mouse brain, I think. So it's a little more difficult. So many engineering and scientists are trying to develop a much more sensitive ways to do the function, the neuronal activity. And in the future, maybe the MEG just designated today and open the facility. That's another possibility. You can directly translate the compare between mouse and the humans. So that's a very important aspect that we're doing.
I know that one of the things that has happened here at the McGovern is putting something in that's missing, or putting back something that was mutated and then creating something. Is that happening in autism, where you're finding something missing, that like the protein or some other gene that you can add back to fix the problem?
Can you maybe talk about some of the rescue experiments?
Yeah, so yes, so there are genetic ways in mice. We can almost do anything. You want, if it's mutated, if we find a human mutation, we can put exactly the same mutation you found in human patient into a mouse, make a mutant mice. You can also put exactly the same gene the non-mutated, the normal gene back in the human, in mice, not only in the mouse, but in the particular brain area to see if I restore the function of this particular area, for example, amygdala, is that enough to rescue the full spectrum or only small aspect? And so this is a completely possible that actually doing. So in the repetitive compulsive behavior, if we actually did an experiment, we put the protein only back in the striatum, that rescues it. Not only that, you let the mouse develop this problem, have lesions, everything. Then in adult, you put them back, it completely cures it. Not only they stop doing this, their skin, everything heals. So it's possible. So it suggests that it's not at least in some cases, they're not a developmental defect that's not irreversible. It's at least irreversible, or it's actually a functional defect that you can. So that brings hope. If not everything studied in mice can be translated in human, but some of it you do. So if this is transferable in human, it gives you hope that this can be corrected in the future in humans. So it's definitely possible.
I mean, this result is truly astonishing. I guess it's probably clear to most of you already, but just to underline, to me, the default assumption before these recent results of Guoping's and a few other related things in other labs was just that if things go wrong during development, well, there may be some symptomatic treatment, some ways that you can teach people to work around the problems, but you don't usually think of things as so changeable in adulthood. So this is a really magnificent and exciting result. Yeah. Sorry, there was another question over here. Yeah.
I have two pieces I'd like to address, and maybe you could speak about it. First of all, I think part of the problem with this facial lack of content is these kids are working so hard just to figure out where they are, so they're really centered on themselves. And secondly, trying to figure out what's going on with their relationship with the world. And if you think about it, we live in a three-dimensional world, but they're actually two-dimensional type people. They're having a hard time spatially fitting in with the environment. So that's one thing. Number two is they've got circular thinking and very poor short-term memory, which prevents them from being social to begin with. So the circular thinking, impedes any kind of creativeness or spontaneity or anything like that. And thirdly, back to the spatiality concept, what I've observed of many kids over the years is the flapping, the hand movement. They're trying to figure out their bodies in relationship to the world, and it's a physical question. I don't know if you follow all this, but this is what I've observed, and I think if you could really get into the circular thinking, I think it would be great, or the repetitiveness, because that's what really impedes them from being at all spontaneous.
I mean, one of the things, important points you're pointing to, is to me one of the real mysteries in autism is which of these different aspects of the disorder are related to each other causally, right? So I think you're suggesting that some of the difficulties with gaze and processing faces may be, in fact, a result of other difficulties. And I think that's a really central question. It's one of the ones that we're trying to investigate in our research program, where we test each kid on an exhausting number of different behavioral tasks to try to figure out which of these things go together kid by kid. And by the way, huge thanks go to I guess there's a few parents here, to those of you who have brought your children in for 2-3 days of testing, which is a huge thing to ask of parents, and we're greatly appreciative of your participation in our studies. We realize it's not a small thing to come in for 3 days of testing. But on that note, I was actually wondering during Guoping's talk whether the two phenomena you reported with the Shank3 mice, the social avoidance and the avoidance of novelty, right on this point, do you think that's one thing or two things, right? Because another mouse or another person is a very complex, mysterious, kind of unpredictable thing, much as a novel object is. And so I wonder whether that's really two distinct phenomena. Can you tell?
We have series, so we're testing them. So one of the most interesting things I have ever seen for autistic kids about the study was eye tracking and a video. For a room of toys and computers and teacher in front, normal kids go in, they will gaze on the teacher and they look at the smiling face and chat with, then start looking what toys I can play. Autistic kids go in with eye tracking, this is our studies, show that they are equally scanning, there's no preference for human face, right? So that got really interesting to me, to many other people. Why would the autistic kids see human face and the toys and the computer are identical? And then it will fix oh, this is the toy I want, and we keep playing it. So one of the theory by Function Mice study, some of the studies suggest that maybe it's also involved in the rewarding system. You know, you interact with people, it's a very rewarding, smiling face. So I think partially it's related. The repetitive behavior is mostly contributed to the dorsal striatum, and the rewarding system is mostly to the ventral. So it's a communication between cortex, at least in part of, from cortex to the striatum, but in a different area. Now, in the behavior assay itself, if they are so interested, restricted interested to do something else, just like you said, whether they are, you know, 4 of them thinking about the other things, they may be less interested in you know, interact with other human beings. So it is possible they are, you know, linked, but I think to a certain degree you can separate them out mechanistically in the brain. That's our hypothesis. We're testing them, at least in mice we're testing them. But the function mice, you guys also testing by different, you know, facial and interactions with them.
Yes, so I just have a comment. So I work on synapses, and as Guoping's picture so elegantly showed, there are lots of things going on at synapses. There are lots of proteins, and the dynamics are incredible. They're positioned as something, you know, singular. But things are turning over at different rates. There's protein synthesis at synapses, and you could potentially get defects at sets of synapses for a variety of reasons. So the real problem with a disease like autism is that it's probably not one thing. It could be, in some cases, one thing that affects a variety of proteins. SAP apps scaffold a lot of the proteins at synapses. And so there are lots of things that could be wrong. And so it's not even clear if we had a single gene that we knew was definitely associated with autism, that we could replace that gene and solve the problem. Because there are lots of regions in the brain that are necessary to function, to work together. So it's a real challenge, and it's still a great unknown. There are lots of us working at synapses now, but we're only just beginning to understand, with things like optogenetics and finer ways of imaging the dynamics of synapses, that it's not just a question of identifying, it's a question of identifying those things that influence things and organize them in time. So this is the challenge on a lot of these diseases. It's probably one of the challenges in schizophrenia too.
I think this is really one of the big challenges. The question is, speak loud. Too loud. Oh, okay. I think that really is one of the problems. I think that there is, ultimately, there will be many, many causes of autism and whatever parts of autism there will be. What I'm interested in is there any research that suggests better or worse ways of treating people who have these many problems and disabilities? I don't hear very much about research in that area. You know, when you have a stroke, people will tell you what area of the brain is causing this, and then you talk about the plasticity of the human brain. It's incredible. But what about the autistic people's plastic brains? I don't hear anybody talking about what you do with the huge numbers of people who are still so troubled. Is there an end point? We have an autistic nephew, and we don't know whether to think he's being lazy often, or whether he's being stubborn. He's sometimes both. But how far do you push it? And it's in that area that I'm most interested in. You know, what is being done to figure out how to best help these people?
Yeah, no, it's a hugely important question. Though this is not the area I work in most closely, my sense of this is that still the standard kind of intensive training methods that have been the kind of norm in autism for, you know, at least 10 or more years are still thought to be the kind of the best shot at the moment. But I think the kind of basic research that Guoping is doing does give hope that you know, changes that happen later in life may be able to kind of continue advancement. I don't know, maybe you guys want to add
So
to that.
I want to emphasize one thing. What we trying to do, I think many of us trying to do, is we now almost clear from human genetic studies, not one or two or even 10 genes are contributing to autism or schizophrenia. There are many, many genes can contribute to that. And each of these mutations may only contribute to a very small percentage of patients. So you have a vast variety of different underlying mutations or factors contributing, combined by environmental factors and other things. But our idea is, for whatever the reason, and autism has core symptoms. If we think about how brain works, for any particular core symptom, must be a common, if the common symptom, there must be a common neurocircuitry activity that control that. So if we understand what a neurocircuitry control a particular behavior, then it doesn't matter what causes it. We can modulate that activity and to treat a variety of different patients with a similar. For example, repetitive compulsive behavior. Your OCD has it, autism has it, Tourette's has it, trichotillomania. And the idea is, if we really figure out that the dorsal striatum is the key, which we have tons of evidence from human studies and from animal studies, then if we find a way to correct that problem, then this medication can be treated for a variety of disorders. That's the idea, that we are not going to replace every single gene, every gene mutation, which is not feasible right now. The idea is understand the neurocircuits defects and find a common way to treat, at least a significant group of them. There may be several different subgroups of them. That's the idea. That's how the basic animal research will help to understand that.
I think that the game-changing approaches to treatment will come out of basic neurobiology, because that's, I also think that people's lives can be made better without game changers. We want the game changers. We have to do the basic neuroscience, but we have people now, right? There's in practically every childhood neurodevelopmental difficulty, there's a shocking absence of random assignment investigations of behavioral effectiveness of treatments. This is true of dyslexia, of ADHD. People are fast for the medications. There's a few widely used behavioral interventions that Nancy mentioned and that you know about. But anything else that might help. You hear a lot about cognitive training or this kind of thing. There's shockingly few reasonably controlled studies of those and I think it's a shame, because maybe those things could make life meaningfully better for people now. But I think physicians, parents, patients have very little information in the way you described it. It's a stunning absence of research.
I think part of the problem is, if you have a kid with autism, you're not going to just volunteer for a random assignment. You're going to do everything you have the resources to do, of course, and so that makes it challenging to do those studies.
So I understand that, of course. But there's I've heard of, there's studies just now, just beginning, amazingly, of things like cognitive training in young adults. Okay, now, does that help also? Is it a waste of their time? That's worth knowing.
It does help many psychiatric problems. Why not?
Yeah, and so where is the study that tells you, it might help and maybe help some kind of a person but not another? And it's just amazing about the absence of evidence about that.
We're getting close to the end of our time for the questioning. Is there someone who had a question who hasn't had a chance to ask it before? Go ahead.
I appreciate the rise, and when you take all the things out, old age, parents, all that stuff, but there's been a rise in autism of some percentage. Do any of you have theories or beliefs in prevention, toxicity? I mean, all our children, BPA in bottles, anything that parents could be doing. Do you have any little gleanings of evidence of something that might be causing this that could be involved in chemicals or toxicity?
And I'm not going to give you the answer that you want to hear, but in my mind, the answer is we don't have those answers. We can't give you that information because the research just doesn't exist yet. There are a lot of environmental factors of interest that are being investigated, but to date, there aren't any that you can point to and say, these are the ones that are really contributing to an increase in autism risk. It's partially due because the research funding isn't there, but also because some of these studies are very difficult to undertake. You hear things of studies requiring large sample sizes, or what's called a longitudinal study, prospectively collecting data. There are some projects that are underway or being planned, one being the National Children's Study that the federal government is funding for the most part, that will track mothers from the time of conception all the way through their pregnancy to birth, and then following those children up to the ages of 21 years old. So that might give us some insight as to what types of environmental exposures might be contributing to autism risk. But it's going to take time to collect that data. So that's the problem. I know that families have these questions now. They want to know what's caused their child's autism, or how can we inform our relatives who are thinking about having children of what to do and what to be careful about. But unfortunately, there's just not that kind of information available yet.
I was thinking of people whose families donate the brains of Alzheimer's. Well, they don't know till they investigate the brain. I understand that, but 21 years to people who are struggling with a loved one with autism sounds. It hurts, it just hurts. But I just thought I'd throw that out because I think most parents are being terrifically careful about toxins now, just from, if you will, commercial, network, casual reading. We know we're not doing the best to our children with toxins. So I just hope that perhaps there'll be something you'll come up with. Thank you.
Reinvestigate the role of vaccines in autism, okay? I'm their developmental neurobiologist. And it's very interesting. This is a committee made up of neurologists, pediatricians, a whole range of scientists going back and looking at all this literature. And one thing, the only thing you can say is that in some instances, in one instance, for example, there's a sodium channel mutation. And if you give somebody measles, mumps, or rubella vaccine, that person can develop epilepsy because they're already susceptible. Okay, and if you generalize that, and I firmly believe this for studying development for many years, there are susceptibilities we're all born with. Right, and so you hear about something in plastic bottles, or so, and it's possible, but it could only be in 0.1% of the people. So you can drive yourself nuts trying to raise somebody in a bubble, and that's probably not good for them too, right? So it in a disease like this, it is so complicated to know what the factors are. And the really only thing, and I don't know if you have statistics on this, but early intervention does help. Early intervention and training does help. Not for everybody, but for many of them.
So we're going to have a reception outside, and that would be a good time, first of all, to just informally talk with some of the speakers and other faculty that are here. But also, if someone would like a tour, an optional tour of the Imaging Center, there'll be someone outside who will raise her hand and just go to her. And
I'm back here. If you're interested in a tour, I'll be waiting for you outside, and I'll take you downstairs.
She can take you down to the right away. Yes, it'll be immediately after this, and then you can go down, take a tour, and come back and join the reception. Thank you all so much, and thank you for coming. Great.